Welcome to your week 7 mini session for ST210, chapter 16, gram-negative bacilli and cocobacilli. Let's get into our chapter objectives. We have to talk about key terms. We have to discuss the mechanisms of disease, identify body systems, look at pathogenicity, identify gram-negative bacilli frequently responsible for HAIs, and then put everything that we thought into critical thinking and how it relates to our environment in the operating room. But with every microbiology, there's always clinical significance. Now, some bacteria in our gut are normally harmless and even helpful, but they can cause serious infections if they enter the body in other parts. Now, we as surgical techs, we use strict bowel technique during gastrointestinal surgeries where we're dealing with the intestines to prevent this risk. This includes isolating any instruments that contact the intestines, using extra drapes around the wound, changing gloves after touching intestinal tissue, and using new sterile instruments for closing the wound. Following these steps, as outlined in the AST guidelines for bowel technique, it helps prevent surgical site infections and ensure safe patient care in the operating room. Now let's get into the chapter. We're going to start off with phylum Proteobacteria. It is a large group of gram-negative bacteria with classes alpha, beta, gamma, delta, epsilon, and zeta, containing some of the most harmful bacteria for humans. When I mean by harmful, when we talk about alpha, we look at rickets. Beta, I want you to think of Bordetella. Gamma, think of things like Salmonella. We get all the way down to epsilon, and we have this bacteria that's really long, Helobacteriaceae, okay? Now, with Brucella, these are small, gram-negative, non-motile bacteria in the alpha class. So they're going to go and they're going to infect the body through breaks in the skin, your mucous membranes, your ingestion of unpasteurized milk, or even just inhaling aerosols. Once inside, they live in the RES, which includes your organs like your liver, your spleen, lymph nodes, and can form granulomas, which are small areas of inflammation. There are 6 species, 4 of which infect humans and cause brucellosis, which is a zoonotic animal to human disease. Now, each species is linked to a specific animal. Now, Brucella can survive in the body by resisting destruction from immune cells. They avoid degranulation, which is the release of toxic substances by our immune cells, and a resistance to phagocytosis, where they're trying to avoid being engulfed and destroyed by our immune system. Kingella. Now, this is a part of the beta class, and it includes species such as Kingella, Kingea, K. dinfrenicans, and K. oralis. These bacteria are gram-negative, they're short, they're also non-motile, and they look like cocci. All right, now, K. kingea is a pathogenic and mainly affects children living in the respiratory and the mouth areas. It can cause serious infections like bacteremia, which is blood infection, osteomyelitis, which is bone infection, and endocarditis, which is heart infection. We also have K. dinfrenicans and K. oralis. They are similar to gonorrhea and can be spread through oral trauma, like aggressive dental cleaning, into where they're getting in the deep tissues and blood, potentially causing endocarditis, which again, remember, that is a heart infection. Chromiobacterium is a genus in the beta class. Now, the bacteria are large. They are curved or rod-shaped. They're gram-negative. They are motile, meaning they're moving, and they can survive with or without oxygen. The main species, C. Vioculum produces a violet pigment and is the only microbe that produces this pigment and causes infections in humans. So these bacteria are not part of the normal human microbiota, but are found in soil, water, and animals. Infections caused by this can have a very high mortality rate due to the production of endotoxins. We get into our Alkanea family. Okay, again, a beta class, which means they share certain traits useful so that we're able to identify them. Now, this bacterium is often found in water, especially in salty, saline environments. It can form biofilm, and remember that that's that slimy layer of bacteria that sticks to surfaces. And in healthcare, it has been found in the inner parts or the lumen of central venous catheters, especially in patients with cystic fibrosis. Now, this bacterium is now a concern in oncology, which are cancer clinics, because it can cause HAIs and emergent issues for our most vulnerable patients. You've heard of Bordetella. It is the most well-known type in this family. They are small. They are gram-negative bacteria that need oxygen to survive. They exist alone or in small groups and are recognized as a mix between round and rod shapes. The three types of Bordetella can cause infections in humans. You have B. pertussis, which is known as whooping cough, mainly affecting infants and young children. This infection can be very serious for infants under 6. We have B. parapertussis, which is a less virulent form but also causes respiratory issues. And then bronchi septicemia can infect humans but is more commonly associated with animals. Now, Bordetella pertussis is the most dangerous of the three because it produces specific proteins and toxins that increases its ability to cause disease. We move on to the Enterobacteriaceae, also known as enteric bacteria or enterics. Mainly consist of gram-negative bacteria that are typically found in the gastrointestinal tracts of humans and other mammals. These bacteria are non-spore forming, they're motile or non-motile, and they grow best at 37 degrees Celsius, which is about 98.6 to 99% Fahrenheit. E. coli is the most well-known species in the Gamma class, and they are gram-negative, rod commonly found in, of course, our intestine tract as normal, harmless parts of our microbiome. Most E. coli strands are actually beneficial, helping with vitamin synthesis for good health. However, some strands can cause gastrointestinal diseases. Now, E. coli can cause various diseases, including urinary tract infections, sepsis, diarrheal disease, and neonatal meningitis. Make sure that you really have a good grasp on the 5 pathogenic strands of E. coli, okay? You have enteropathogenic E. coli, EPEC, enterotoxigenic E. coli, ETEC, enterohemorrhagic E. coli, EHEC, enteroagglutinative E. coli, AEC, and enteroinvasive E. coli, EIEC. These strains cause diseases through different mechanisms, often linked to their ability to produce toxins or invade the intestines. Now, other things to think of is that these infections typically begin with lab cultures, which are essential for identifying the bacteria. 2 sets of culture specimens should be collected, one aerobic and one anaerobic, ideally 20 to 30 minutes apart and from 2 different sites. There are other tests that may include blood and urine studies, imaging such as X-ray, CT, MRI. You may have what you see over here, an endoscopic retrograde colonopactography or ERCP, to check for biliary obstruction. Sometimes we can do a needle biopsy of soft tissue, surgical drains, or even, as you see here, a lumbar puncture for cerebral spinal fluid. Okay. Now, treatment is always going to involve some type of antibiotic therapy, but these bacteria can unfortunately develop antibiotic resistance quickly, making treatment so challenging. Now, we also look at Klebsiella, which is non-motile, rod-shaped, gram-negative. They make things resistant to the body's defense and many antibiotics. So some of these include pneumonia, which is a common in health care associated infections, particularly in respiratory and urinary tracts. We have oxytoccia and granulomatous. Now, these infections, of course, cause pneumonia or bacterial septicemia, cholecystitis, diarrhea, meningitis, osteomyelitis, and are also linked to less common infections like granuloma. So just keep that in mind. Now, diagnosing infections often require, again, lab cultures. We have Citrobacter species, which are gram-negative bacteria that can survive by using citrate as their main energy source. These bacteria are extremely opportunistic, meaning they often infect patients that are already weakened and have an immune system that is not as strong. They can cause severe infections like UTIs, neonatal meningitis, and respiratory infections. Common species include K. corseri and C. frendui. Gotta love these terms, right? There's also gram-negative bacteria, and they are part of this family. They are commonly found, again, you're going to hear this a lot, in the gastrointestinal tracts of humans and some animals. We look at Providicea. Now, this species mainly causes infections in the urinary system, but can also lead to gastroenteritis and bacteremia. A common species is often found in animals such as cats, dogs, birds, sheep, and reptiles, where it usually lives harmlessly, but can unfortunately become infectious in us as humans. It is an opportunistic gram-negative bacterium. It's known for producing a distinctive red-pink pigment and can cause a pink-colored breast milk in cases of postpartum mastitis. Okay, now this pigment has immune suppressive and anti-tumor effects, adding complexity to infections in immune compromised patients. Shigella, another one. Now this genus includes gram-negative, again non-motile but non-spore forming rod-shaped bacteria known for causing shigellosis. There are 4 primary species. You have group A, often causes severe outbreaks in areas with poor sanitation. Group B, group C, and group D also causes different severity of the diseases. Okay, so one you heard of, Salmonella, gram-negative, rod-shaped, motile bacterium with over 2,500 servers, many of which are pathogenic to humans. We have Salmonella typhi, causes typhoid fever through fecal oral transmission, basically contaminated food or water. It survives within macrophages. It spreads through the circulatory system and can cause a rash. With post-recovery, some individuals may become chronic carriers. And there was one famously known, Typhoid Mary. Okay, if you ever have time to look that up, it's very interesting. We also have Salmonella typhimurium, frequently associated again with foodborne outbreaks, but in large-scale meat processing plants. And Salmonella enteritis, known for outbreaks linked to egg contamination during packaging. Yersinia, This is the final genus in this family with significant medical importance. Now, Yersinia pestis, which is the gram-negative, it's anaerobe associated with the plague, bubonic, septicemic, and pneumonic. So it's 3 types of plagues. It's known for bipolar staining and slime production due to a capsular antigen. It causes bubonic plague with swollen lymph nodes called bulbs that become dark and hemorrhagic, leading to that name, famously Black Death. So we look at the disease progression. Infection can rapidly spread through lymph nodes, the lungs, the spleen, liver. Pneumonic plague results in severe bacterial pneumonia, making the disease highly contagious as infected individuals expel bacteria through coughing. Untreated infections have a high mortality rate with death possible within 24 hours after symptoms. So to treat it, it involves sputum, blood cultures, and even lymph node biopsies. Effective treatment includes gentamicin, streptomycin, and tetracycline. Now, control efforts historically focus on eradicating rat fleas, which is the primary plague vector. We get into Morolaxicae family, belonging to Gamma. This family includes cocci and bacilli bacteria. They contain species that morphology from like a gram-negative. So they're non-motile aerobes that are found in the soil. Now in healthcare, this accounts for approximately 80% of HAIs with this particular genus. The species thrives in fluid environments, but is also capable of surviving on human skin and dry surfaces in hospital. Common in the ICU, it often colonizes hospital irrigation and intravenous solutions. So strict hand hygiene is important in preventing, especially the spread, okay? One of the things to think about is you may be doing a tracheostomy, which is a opening or placing a hole in the trachea, which is where you're allowing direct access to that trachea for breathing support, which can be a site for this particular bacteria colonization in hospitalized patients. We get into Pathogenicity family. Now, they can survive with or without oxygen, but they need enriched media to grow in labs due to their particular nature. You have heard of Louis Pasteur, right? Now, Pasteurella bacteria are often found in the mouths and throats of animals as harmless residents, but can become infectious, which is commonly linked to animal bites, growing easily on the blood or a chocolate agar, and produces a foul smell. It can cause local wound infections, respiratory infections, blood infections like septicemia, and even, again, here's that word again, endocarditis, which is a heart infection. We also have the Vibronicae family. They're members of the Gamma class. These gram-negative, rod-shaped bacteria may be straight or curved and often have flagella for motility. Now, they include species that produce extracellular cytotoxins and enzymes, causing significant tissue damage and infection. We have the Vibrio diseases categorized into chloria and non-chloria forms. The biggest thing to keep in mind is that when we look at historical outbreaks, they have been noted with water and humans serving as primary reservoirs. Your stomach acid generally will neutralize the V. chloria, but individuals with low stomach acidity due to conditions like a gastric surgery, a vagotomy, they have H2 blocker therapy, have a heightened infection risk. So just keep that in mind. So if you're in those type of cases. We also talk about the vagotomy, a surgical procedure involving the cutting of the vagus nerve to reduce stomach acid production, which may increase susceptibility to infections like cholera. We have the genus Aeromonas, was originally grouped with the Vibronicae family, but due to just different studies, it's now its own within the Gamma class. Now, these bacteria are gram-negative, rod-shaped, can survive with or without oxygen. The species are found in various environments, including fresh and saltwater soil, sewage, and some foods. This widespread presence makes them common environmental bacteria with potential to cause infections, especially in those who are already immune compromised individuals. We get to family Pseudomonaceae. They are part of the Gamma class. It includes Pseudomonas, which is a group of non-fermenting gram-negative bacteria. These bacteria are rod-shaped, non-spore forming, and motile. They're moving with a polar flagella. They can produce a slime layer that helps them stick to surfaces, enhancing their ability to cause infections and resist treatment. They have a sturdy, impermeable cell wall and enzymes that break down toxins, making them tough enough to survive in some of the most harshest conditions. We look at how they are common in hospital settings. They are thriving in sinks, soaps, humidifiers, and fountains due to minimal nutrition requirements. Healthy individuals can develop ear or skin infections from improperly chlorinated pools or hot tubs, just to think about. Or let's say you wear contact lenses for a long time, you can get conjunctivitis. For our immune compromised patients, they have a high risk for severe infections, especially in those with cystic fibrosis, with burns, leukemia, or those on a ventilator or with tracheostomies. Our family Campylobacteriaceae. This family includes, again, gram-negative, non-fermentative bacteria that are spiral shaped. They are motile rods with unipolar or bipolar flagellas. Now, the most commonly isolated species, which is the Campylobacter gingi, they're highly sensitive to the environment and unable to survive in 21% oxygen, dry, or acidic conditions. It grows optimally in conditions with 3 to 5% oxygen and 2 to 10% carbon dioxide. It is commonly found in birds without signs of infection and can be transmitted to humans through contaminated chicken or manure contaminated unpasteurized milk. Now, C. gingi is a leading cause of diarrheal issues or illnesses in the United States, surpassing Salmonella. We get our family Helicobacteriaceae. This includes the Helicobacter genus, known for its similarities to Campylobacter. Now, these are gram-negative. They are microphilic bacteria, meaning they need low oxygen levels, with a curved shape and motility, and may test positive for catalysts. The most notable species in this group is H. pylori, which is primarily responsible for causing chronic gastric and stomach and peptic ulcers. It can survive in stomach acid environments by colonizing the gastric mucosa, which is our stomach lining. And then over time, infection with H. pylori can lead to persistent inflammation and may even contribute to the development of stomach cancer. The last part we're going to talk about is our bacteroides. Now, part of the bacteroides phylum, these species are obligate anaerobes and make up a significant part of the normal human digestive tract microbiome. Now, although most species are harmless, some can, as you've learned, cause infections. Bacteroides fragilis are small, they're gram-negative bacterium that grow on blood agar. It is a non-spore forming and non-motile, but it can cause abscesses due to its large capsule, which resists immune attacks. It's associated with peritoneal and pelvic abscesses, bacteremia, and diarrhea in people with inflammatory bowel diseases. So, This has been your mini session, Chapter 16, Gram-negative bacilli and coccobacilli. If you have any questions, any concerns, please make sure to reach out, and I look forward to seeing everyone who can attend the live session.